woman with blood type O and a man with blood type AB have a child together. What blood types are possible for their offspring? Now, most students might think that the best way to approach this problem is to just memorize the blood type charts and the patterns that they have. But this question is really testing whether you understand Mendelian genetics. Because if you understand how alleles are inherited, how dominance and recessiveness works, and what codominance is, we can solve this entire problem systematically instead of by memory. And by the end of this video, we're going to come back to this exact problem and solve it step by step. In this video, we're going to cover alleles, dominance, recessiveness, codominance, Punnett squares, and blood type inheritance, so we can solve genetic questions systematically. So Mendelian genetics exists because traits are inherited in predictable patterns called genes and alleles. So a gene is just a unit of heredity, and it codes for a trait. Now, linked to a gene, we have alleles. Alleles are different versions of that gene. For example, a specific gene may code for eye color, and the alleles of that gene would either be blue, brown, hazel, or a different type of eye color. So they're all coding for the same thing, however, it's a different variation. Alleles of an offspring are given from both parents. Each parent gives one allele to an offspring. So the total will be 2 alleles for that specific gene. Your genotype is simply the allele combination you carry, whereas the phenotype is what is actually observed. So these two ideas are very similar and they are related, however, there is a clear distinction between them. And the phenotype, which is what you can observe physically, does not always reflect the specific genotype that's associated with it. But for the most part, whatever genotype you have will give you the expected phenotype. Alleles are categorized as being dominant or recessive. A dominant allele is shown with a capital letter. In this case, I'll give it a capital A. The recessive allele will be given a lowercase. So in this case, it's a lowercase a. Like we mentioned earlier, your genotype is going to be the product of one allele from each of your parents. In a monohybrid cross, we're going to have 2 alleles, one from each parent. We can either have a dominant allele from each parent, 1 dominant and 1 recessive, or 2 recessive. If we have a dominant allele, regardless of what the second allele is, we will show the dominant phenotype. If we only have recessive alleles with no dominance at all, we will show the recessive phenotype. That means that both of these combinations will give us the dominant phenotype, whereas only our lowercase a gives us the recessive phenotype. This means that someone can carry a recessive allele and not show the recessive trait. This type of individual is called heterozygous. Whereas if you have 2 of the same, so either 2 capitals or 2 lowercases, you are called homozygous. And to differentiate, you would either be homozygous dominant or homozygous recessive. Now we need to understand gametes. Your body cells are diploid, meaning that they carry 2 alleles for each gene. But sperm and egg cells, or your sex cells, are going to be haploid, and they only carry 1 allele. During meiosis, the 2 alleles separate so that each gamete only receives 1. So we may go from a heterozygous capital A lowercase a to a split where one gamete has a capital A and then the other one has a lowercase a. And then only one of those will end up being fertilized. This concept is Mendel's first law, or the law of segregation. In this idea, when an egg is fertilized from a sperm, the 1 allele from the egg and the 1 allele from the sperm will come together and form that diploid gene combination for the offspring. Punnett squares are a great visual way to organize all the possible allele combinations we can have. So in a monohybrid cross, mono meaning one, we will have a 2x2 graph being made. And let's say we have two heterozygous individuals mating in this case. What we'll do here is we will just look at the respective column and row and combine which alleles there are. So in the top left, we'll have capital A, capital A. Moving on to the right, we'll have a capital A, lowercase a. Same thing on the bottom left. And then on the bottom right, we'll have lowercase a, lowercase a. This is a way we can see that if we cross two heterozygous organisms, we will have a 25% chance of a homozygous dominant genotype, 50% chance of heterozygous genotype, and again 25% chance of a homozygous recessive genotype. Like we mentioned earlier, this does not accurately reflect the ratio for phenotype. So while our genotype is a 1-to-1 ratio, our phenotype is going to be a 3-to-1 ratio, because 3 of our 4 combinations have at least one dominant allele. So we will see a 3-to-1 ratio. Now, crosses aren't only monohybrid, meaning just one allele pair with another allele pair. We can have multiple. Commonly, we will see a dihybrid cross. So instead of seeing 2x2, so we can write it instead as AA, BB, and then we can do it again, AA and BB. So we will see instead of having a 2x2, we're going to end up with a 4x4, giving us 16 total combinations to write here. When we do this, we will end up with a combination of 9, 3, 3, 1. This ratio means 9 both dominant traits, 3 one dominant, the other one recessive, 3 going the other way, 1 dominant and 1 recessive, and then only 1 is going to be fully recessive. Again, this is only if we have a heterozygous cross. Just as an extra note, if we wanted to go backwards, where we see that there's a dominant phenotype, but we don't know exactly what the genotype is, that's where we're going to do a test cross. So in this case, this can be for both monohybrid, so a 2x2, or a dihybrid, or even further. We will cross our unknown dominant organism with a fully recessive organism. So if we look at it from a monohybrid cross perspective, we will not be able to do the Punnett square here because we don't know what alleles will be with this unknown organism. However, once we mate it with a fully recessive organism, we should either have a 50% dominant and 50% recessive outcome, or a 100% dominant, 0% recessive. Let's break down what that actually means. So if we are 100 and 0, that means that our allele combination has to be fully dominant, homozygous dominant. And that's because when we're crossing with a fully recessive, every single recessive is going to be paired up with a dominant, and every dominant is going to be paired up with a dominant. So regardless, we're going to show a dominant phenotype. If we have 50-50, that will be a heterozygous individual, and when we're crossing it with a homozygous recessive, we'll get 50% of our outcomes match with this capital A, and the other 50% match with this lowercase a. Now let's look at codominance and also compare it to incomplete dominance. These topics are usually taught simultaneously, but they have different implications. Codominance, we see that prefix of co. That means alongside. Codominance means we're going to have the two alleles expressing simultaneously. So if we have a heterozygous individual, let's say this is going to be for a flower. If capital A is pink petals and lowercase a are white petals, when a fully dominant and a fully recessive flower cross, so one completely pink, one completely white, the outcome is going to be 100% heterozygous, and we will see white and pink flowers happening at the same time. So this is not going to be a blend, it's going to be both separate phenotypes being expressed together. Now, this is where a lot of people get confused between codominance and incomplete dominance. Incomplete dominance is just that there is a blend and the one allele doesn't fully mask the other one. So if we had a similar scenario, except we had red flowers for capital A, and then again white flowers for lowercase a, we would not see some red, some white like with codominance, but instead we would see them all become pink, because neither the white nor the red are going to fully dominate or take over the other one, and we're going to see a blend of both of them. Now let's move into blood types, which is going to be the main topic going back to that first question. In blood, we have the ABO system. Blood types are determined by the antigens on the surface of the red blood cells. Antigens are just molecular markers that are on the outside of a cell. If you have A antigens on your blood cells, your blood type is A. Same thing with B. If you have B antigens, you are blood type B. If you have both A and B antigens, you are type AB. And if you have neither antigen, you are type O. So now let's see how this relates to genetics. So we have I to the A, I to the B, and then just a lowercase I. The IA allele codes for the A antigen. The IB allele codes for the B antigen. The lowercase I, however, is a little bit different because, like we said, it does not produce an antigen. Now, IA means you have type A blood. IB gives you type B blood, and the lowercase I is for O blood. And that's what makes O the universal donor. The I for A and B are both written as an uppercase, and that's because those blood types are dominant to O, which is recessive. That's why it has the lowercase I. Now, the relationship between these two capital I's is that they are codominant. If we were to make a Punnett square with the two parents, again, a monohybrid cross, let's say the one parent is IB and lowercase I, and then the other one is IA and lowercase I. Well, let's look at what we can have. We can have IA, IB, IA with a lowercase I, IB with a lowercase I, and then two lowercase i's. How can we apply this to the scenario? Well, since these two are codominant, the IA and IB, this correlates to blood type AB. If we have IA and a lowercase I, that lowercase I means it's recessive. This is going to be type A blood. The same thing's happening here with the B and the lowercase I, where we get B blood. And then if we're not producing any antigens here at all, we're going to be type O. Now, if you want to look at if you're going to be positive or negative, that's going to be adding another step to this cross. Your immune system makes antibodies. And this is to protect against antigens that you do not have. So if you are type A, you are going to have anti-B antibodies. Similarly, if we're type B, we're going to have anti-A antibodies. O is the universal donor, and it can go anywhere because it doesn't have A antigens or B antigens that these antibodies will try to attack. And then we have type O. O has neither antigen, so it creates both anti-A and B antibodies. So the antibodies are looking for the opposite antigen because that's what you do not have. Since O does not have any antigens, it's the universal donor, because the anti-B antibodies won't attack it since there's no B antigens, and the same thing goes for A. This is also why O cannot accept blood types from anyone else that's not O, because it produces both anti-A and anti-B antibodies. So if it sees either antigen, it will attack it. And then we have blood type AB. This is why AB is the universal acceptor. Since it produces both A and B antigens, it has no antibodies. Since it has no antibodies, it's not going to attack regardless of what blood type is entering. With all of this in mind, let's go back to the original problem and solve it completely. So we have a woman with blood type O. We established that blood type O is going to be lowercase I. And a man has blood type AB. We said AB is going to be capital I with an A and capital I with a B, because they are codominant, so they're going to exist together. Let's make the Punnett square based off of this. So we will have IA and IB vertically, and then we will have the two lowercase i's horizontally. So let's see what our cross will be. We will either have capital IA lowercase I, the exact same thing again, and then the exact same thing but with B. So capital IB lowercase I, and then the identical combination again. So let's see what our total combinations will be. We have a 50% chance of having type A blood, since we have a IA with a lowercase I. That lowercase I is from the O, and it's going to be overpowered by the A. So that is 50% for A. And then we have the exact same thing with B. IB is going to overpower the lowercase I, so that's 50% B. Although the mother is type O and the father is type AB, we will see neither of those in the offspring. We can only have type A or type B explicitly. If you want to see more biology topics, the playlist should be on your screen now. Thanks for watching.